Okay, so welcome to this next video in which we are going to discuss intrinsic drug resistance in a species of bacterium known as Pseudomonas Aeruginosa, which is a horrible gram-negative bacterium which is resistant to pretty much every antibiotic that you can think of, nearly. And it's a bacterium which colonizes the lungs, the surface of the lungs in people with cystic fibrosis, and it leads to destruction of the airways known as bronchiectasis. So it's a horrible, horrible infection if you get it, and it's really intractable, basically, infection that doesn't respond to antibiotics and doesn't really is never really beaten by the immune system. Okay, so the first thing I want to explain to you is what's the difference between intrinsic resistance to drugs and acquired resistance to drugs. So intrinsic resistance to the drug really means that the bacterium is not doing anything special to resist the drug. The drug just does not work on the bacterium. It means basically that something in the bacterial cell's physiology just stops, does not allow the drug to function, basically. So the bacterium doesn't have to alter its physiology in order to resist the drug. Whereas acquired resistance means that the bacteria has had to change from its normal physiology in order to resist the drug, basically. So, what sort of mechanisms could give a bacterium intrinsic resistance to a drug? Well, potentially the target just would not be present. If the target isn't present, then the drug's not going to work. Target not present. So, for instance, the antibiotic isoniazid. So let me give you an example. Isoniazid, which is used to attack tuberculosis, Mycobacterium tuberculosis, it stops the synthesis of mycolic acid, basically. So that's very powerful in mycobacterial species. So this is very powerful in Mycobacterium tuberculosis because Mycobacterium tuberculosis has a membrane made up of mycolic acid. Okay, so if you can stop it synthesizing that, then you're going to lead to damage of this bacterium. However, if you use this drug against a bacterium that does not synthesize mycolic acid, then that isn't going to have any effect because the target just is not there, basically. Okay. Another way in which you could get intrinsic resistance is if you just did not allow the drug to get into your cell, basically. What if the drug just could not pass through the cell membrane? So permeability problems. Okay, so if the drug just cannot actually get into the cell, if it just can't get to the target, so the target's present in this case, but it just can't get to the target, then that would give you intrinsic resistance. We're going to see that one of the mechanisms of intrinsic resistance in Pseudomonas aeruginosa is indeed that its outer membrane just isn't permeable at all, really, to our drugs. It's very difficult to get our drugs to go through that membrane. Okay, and other examples of how you could achieve intrinsic resistance is if you have transporters that are used anyway for physiological purposes. So if you've got these transporters which are working physiologically anyway, and then it just so happens that they efflux the drug. So transporters which efflux the drug, but they have, I want to stress, they are not put there specifically to move the drug out. They are there just doing a physiological role, basically. So intrinsic means that something in the physiology of the bacterium just means that the antibiotic just does not work. Whereas acquired means that the bacteria has to change its physiology in order to resist the drug. So making beta-lactamases would be an example. So making enzymes that actually break down the drug would be an example of acquired resistance, or completely changing your enzyme so that it's not sensitive to the drug anymore, in the case of MRSA, where it changes its peptidoglycan transpeptidase enzyme so that the beta-lactamases just don't affect it anymore. Okay, those would all be examples of acquired resistance. Pseudomonas aeruginosa, though, is the king of intrinsic resistance. So Pseudomonas aeruginosa is a gram-negative bacterium, which means that it has two membranes, basically, an inner membrane and an outer membrane. So let me draw it here. So here is its outer membrane, let's say, and here is its inner membrane. And if you were going to call one of the membranes its cell membrane, you'd call the inner membrane its cell membrane. So this is the inner or cell membrane, so inner slash cell membrane, okay? And then we have the outer membrane here, so this is the outer membrane. Okay, and in between the inner membrane and the outer membrane there is a space known as the periplasmic space. So this here is the periplasmic space. Okay, and in this periplasmic space there is the cell wall of the Pseudomonas aeruginosa. Now, Pseudomonas is a gram-negative bacterium, which is why it has these two cell membranes, and that also means that it only has a thin cell wall. So this in red here denotes this thin cell wall here. Okay, right. So there we go. Right, so firstly then, the first reason that Pseudomonas is very intrinsically resistant to our antibiotics is that this outer membrane is extremely impermeable. So this is impermeable. It's very, very difficult to get drugs to go through that outer membrane. Usually, the way we get drugs to go through outer membranes of gram-negative bacteria is that the gram-negative bacterium would have porins in its membrane, basically, which we can get the drugs to go through. These are effectively just tunnels through which molecules can pass. But Pseudomonas has very narrow porins that are very difficult to get drugs to go through. Okay, so this is a porin here. Okay, so the outer membrane is very impermeable, and that just means that the drugs just can't get into the bacterium. In addition, it has a massive great transporter which spans both membranes. So it spans the inner membrane and the outer membrane. And this transporter is known as the MEX-AB OPA-RM transporter. So the MEX-AB OPA-M transporter. So this is an example of an RND transporter. Okay? So it's a big transporter which spans both of these membranes. So RND transporters generally tend to span both membranes. So this is an RND transporter, which stands for resistance, nodulation, and cell division. Resistance, nodulation, cell division transporter. Okay, and this transporter performs valuable physiological roles in the Pseudomonas bacterium. It's just basically coincidence that it happens also to transport drug molecules. It plays a physiological role. It's not just put in there because it transports drugs out. It also transports all sorts of other important molecules. Okay, now this transporter is going to couple the movement of a drug molecule out, or any other molecule out, to the movement of protons back across the membrane into the cytoplasm of the pseudomonal cell. So, across the inner membrane of across the cell membrane of bacteria, there is a proton gradient, much like there is a proton gradient across the membrane of the inner mitochondrial membrane. So you have very high proton concentration in the periplasmic space and a low proton concentration in the cytoplasm. Okay? Right, and you also have an electrical potential difference across this membrane, where the electrical potential inside in the cytoplasm is lower than the electrical potential in the periplasm. Protons have a positive charge, so they want to be in the compartment with the lower electrical potential. So the electrical driving force is favouring the movement of protons in. In addition, the concentration gradient is driving protons in. So we say the electrochemical gradient faces inwards, basically. So we will couple the movement of our substance out to the movement of a proton from the periplasm to the cytoplasm, and that is how we'll get the energy to move our drug out, basically. The energy that's released by the proton when it goes down its electrochemical gradient will allow us to move the drug out. Okay, so this is the MEX-AB OPA-M transporter here in green. and basically it uses this secondary active transport mechanism to extrude a huge array of drugs. So it extrudes the beta-lactams, so not just the penicillins, but all the carbapenems, the cephalosporins, the monobactams, they're all transported out. It extrudes the tetracyclines, okay, so doxycycline, tetracycline, minocycline, they all get transported out. It transports out novobiacin, the coumarin, the aminocoumarin. Okay, it transports out the quinolones, so ciprofloxacin, levofloxacin, gatifloxacin, all of those are moved out. It also transports out the macrolides, so the macrolides are moved out as well. Macrolides, so erythromycin, clarithromycin, azithromycin, all of the thromycins, and finally it also transports out chloramphenicol, also called chlornitromycin. Chloramphenicol. Oh dear, squashed in there. Chloramphenicol down there. So it transports out a huge number of drugs, which means that if our drug molecules finally do fight their way into the cytoplasm through the outer membrane, then through the cell wall, then through the inner membrane, they're going to be chucked out again. And this is why Pseudomonas aeruginosa is so resistant to our antibiotics, why it's so difficult to destroy this infection with antibiotic chemotherapy, because firstly its permeability of its outer membrane is so bad, and it has this transporter, the MEXAB-OPA-M, which just basically transports all of our drugs out, and all of the Pseudomonas aeruginosa bacterium have these transporters because they perform a valuable physiological function.